Greetings to everyone. Welcome to this educational channel on biology. In this video, we'll study the importance of nucleic acids in protein synthesis. This subtopic is from the Form 4 KSSM Biology syllabus, Chapter 4, and the subtopic is 4.5 on nucleic acids. So examples of nucleic acids are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Now, these nucleic acids are very important because they have the genetic codes or information for the production of different types of proteins. So the body needs proteins, right, to form the structural components of body tissues, such as muscle, hair, bones, nails, and skin, right, that contain a lot of different types of proteins. And on top of that, proteins are used to form enzymes, antibodies, hormones, and hemoglobin. So proteins are very important in the body and they have many different functions. How does the cell know how to produce all these different types of proteins, which are complex organic compounds? Let's find out more. The learning standards for this video are as follows. We should be able to justify the importance of nucleic acids in cells as the carriers of hereditary information. So hereditary information means information that can be passed down from the parent to the offspring, or parent to child. Now this information is found in the chromosomes, and these chromosomes are passed down from parent to child. Then secondly, the importance of nucleic acids in the production of proteins. We should be able to explain the importance of nucleic acids in the production of proteins. Okay, the importance of DNA and mRNA in the production of proteins as seen here. So before we go into the lesson, let us have an overview of the whole chapter of this chapter 4, chemical composition in a cell. So in this chapter, we have discussed the importance of water, and we've discussed the facts about water, carbohydrates, proteins, and lipids. And so the last one are the nucleic acids, right? So we have already discussed the two types of nucleic acids, which are DNA and RNA. What is their basic structure? Both of them are formed from the basic unit called nucleotide, as shown below here. And each nucleotide consists of 3 parts. Normally, we draw a circle to represent the phosphate group. Then we have the 5-sided carbon molecule. Okay, 5-sided molecule with carbon in it. And that's the sugar. It's called the pentose sugar. All right. And then lastly, we have the nitrogenous base. Okay, B for nitrogenous base, for the base. So P for phosphate group, S for sugar, and B for base. All right. And then for the bases, each nucleotide can have either one of 4 bases. All right. So the base can be, now for DNA, the base can be adenine. So adenine can be found here, or it could be guanine, G, guanine, and adenine is A, okay? Then cytosine, C for cytosine, or T for thymine. So we have to remember these 4 bases that are found in the nucleotides of DNA. So why are there so many different types of nucleotides with different nitrogenous bases for DNA? Why don't you just have adenine? The reason is because the bases form a code, a genetic code that helps the cell to make proteins. So that's why there are 4 bases. So the combination of bases, the base sequence in the DNA. So the bases are found in the center part of DNA here. And the base sequence, for example, first adenine, then cytosine, then after that maybe thymine. Now the base sequence is a code to help the cell know what amino acids to join together. The base sequences is the code for an amino acid. 3 bases form the code for 1 amino acid. So that's why you have different bases, okay, so that the code can be formed. If you have only one base like A, how to form a code? A all the way down, right now. So you must have, here there are 4 different bases, and the combination and sequence of the bases forms the code for the different codes for 20 amino acids that we need to make the proteins in our body. Okay, don't worry about that. We'll find out more as we go along. Now for RNA, it also has nitrogenous bases because its structure is also based on this basic unit, nucleotides. It's made up of a long strand of nucleotides, right? So the bases are quite similar to DNA except for one. So nitrogenous bases in RNA are adenine A, guanine G, cytosine C, and then instead of thymine, okay, there's no thymine in RNA, it has uracil, U. Alright, so we can say that thymine in DNA is replaced by uracil in RNA, for RNA. So this is like something that you can remember as a whole sentence. Thymine in DNA is replaced by uracil in RNA. There's no thymine in RNA, just uracil instead. Okay, that's the replacement for thymine. So another fact we need to know is that RNA is a single strand of nucleotides. So we call it a single strand of polynucleotide. Poly means many. Many nucleotide units join together to form a long single strand. But DNA is different because it's made up of 2 strands of nucleotides. So 2 strands of polynucleotides. Now this is the first strand here, and this is the second strand, and the 2 strands are wound around each other. Okay, to form the double helix structure of DNA. Okay, so DNA has a double because it has 2 strands, helix because it's a spiral shape, right, structure. But RNA doesn't have the double helix structure because it has only 1 single strand of polynucleotides. All right. So firstly, we are going to revise the structure and location of DNA and the genes in a cell. So this picture shows us a cell, right? And in the cell, there is a nucleus. So in the nucleus, there are many chromosomes, all right? Now, this is the structure of 1 chromosome that has duplicated to form 2 sister chromatids, right? And this center part here is the centromere. Now, if we look at 1 sister chromatid or 1 chromatid, it's actually a chromosome, right? So it's made up of a long strand, very long strand of DNA, deoxyribonucleic acid. And this DNA is wound around proteins called histones, all right, which we will study later. So the important component here is the DNA. Now, where are the genes found on the DNA? So the genes are segments of DNA. Segments of DNA form a gene. And genes are used to determine the characteristics of an organism. Actually, the gene consists of a sequence of bases in the center part of the DNA molecule here. And there are 4 bases, adenine, thymine, cytosine, and guanine, A, T, C, G. So these 4 bases are arranged in a specific way. For example, maybe in one part of this gene, we have the sequence of bases A, T, G, let's say. Okay, then this will stand for a amino acid. There are about 20 amino acids that are needed to make the proteins. So a code that is made up of 3 bases, all right, stands for 1 amino acid that needs to be joined together with the others in the right sequence to form the protein molecule. That is the code that is found in the gene. All right, the code is in the sequence of bases, and this sequence of bases contain the information about what types of amino acids to join together to form the specific protein molecule. All right, let's find out more about it. Okay. So the second section is to discuss the role, the function, the role or importance of DNA, deoxyribonucleic acid, in the cell. Our first learning outcome is that we need to be able to justify the importance of nucleic acids in a cell. This means to explain the roles or functions or importance of the nucleic acids, which are DNA and RNA, in a cell. Okay, so let's talk about the role of DNA first. Now, the roles of DNA, there are 3 roles or 3 functions. 3 points concerning its importance. The first one, DNA contains genes with genetic codes or instructions for the synthesis of polypeptides and proteins. DNA contains genes with genetic codes or genetic instructions for the synthesis of these polypeptides here that will then be used to form the proteins. So where is the genetic code or instructions in DNA? It is in the sequence of nitrogenous bases in the center part of the DNA molecule. And the nitrogenous bases that are used are adenine, thymine, guanine, and cytosine, A, T, G, and C. So the sequence of these bases tells the cell what amino acids to join together to form the polypeptide chain. Okay, now we'll discuss more about the code later on. So next, DNA. Now, how does DNA, the genetic code in DNA, lead to the formation of proteins? Okay, so first of all, this code in the DNA has to be copied onto a messenger RNA through the process of transcription. Okay, so transcription is a process whereby the genetic code on DNA is copied onto mRNA. All right, so mRNA strand is shorter and is only one-stranded. All right, a strand of polynucleotide, a strand of polynucleotides, correct? With the bases here, okay, sticking out here. So it copies the code from DNA. Okay, the mRNA copy the code from DNA, and then it will come out from the nucleus to the ribosome in the cytoplasm. Then after that, the mRNA is attached to the ribosome, and then the process of protein synthesis starts. Okay, whereby the code on the mRNA is read and used to determine what amino acids should be brought to the ribosome, should be joined together in the right sequence. So the process whereby, so translation is the process whereby the code on the mRNA is used or is read to determine the types and the sequence of amino acids that needs to be joined together to form the polypeptide chain. That means the code has been translated, the code on mRNA is translated to form a sequence of amino acids that are joined together to form the polypeptide chain. So it's very important that the amino acids used are the right ones, and that the sequence of amino acids here is the correct sequence. Okay, even if just one amino acid is the wrong one, even if there's just one mistake with one amino acid, the protein may be non-functional. Okay, cannot help to carry out its function. Okay, so okay, now come to the production of proteins. What substances in our bodies are made from these proteins? Okay, so proteins are used to make enzymes, for example, digestive enzymes in the stomach, such as pepsin, and enzymes in the small intestine like lipase, amylase, trypsin. Okay, they are all made from the proteins that are formed from the genetic code in DNA by using the genetic code in DNA. And other proteins are muscle proteins like actin and myosin, all right? And also hemoglobin, which is a complex protein in the red blood cells that's used to transport oxygen from the lungs to the cells. Okay, so these are all very important proteins that are formed. Now, so after the proteins are formed, now next, the second role of DNA is that it is a determinant of characteristics in living organisms, meaning it determines the characteristics in living organisms. Like for humans, the characteristics that are determined by DNA are blood type, eye color, hair color, height. Okay, so how are these characteristics determined by DNA? You see, the genes on DNA, all right, have the instructions for the production of proteins, right? So the genes in DNA determine what type of proteins are produced by a person's body. And after that, the proteins are the ones that determine the characteristics of organisms. Okay, let's take, for example, a characteristic or trait such as blood group. How is blood group determined? Now we know there are 4 blood groups, A, B, AB, and O. So for example, a person who has blood group A, all right, has a special substance, a chemical substance called antigen A on the surface of his or her red blood cells. So this antigen A is produced by people with blood group A. So blood group A people can produce an enzyme, which is a protein, that leads to the formation of antigen A. Okay, and then blood group B, they have an enzyme which leads to the formation of antigen B. Okay, so that's how the proteins formed can determine the characteristics of organisms, like blood group in humans. Right, we'll study more of this in another chapter, the how is blood group determined. Okay, so I'm just giving you an example, but that the proteins can determine characteristics of organisms. Okay, now the third function. Okay, so the second function was DNA is a determinant of characteristics in living organisms, such as blood group, eye color, height, etc. Okay, through the proteins that are produced. Now, third one, DNA is a carrier of hereditary information. Let's look at the top here, the left-hand corner here. So DNA is a carrier of hereditary information. Hereditary information means information that's passed down from parent to offspring. Okay, can be passed down. So how is this information, that is DNA, how is it passed down? It's passed down in the chromosomes, right? So father produces sperms, and in the sperm, the most important part is the nucleus that contains the chromosomes, which carry the DNA, the genetic information, for the next generation, for the offspring. And then the ovum from the mother also has a nucleus where there are chromosomes carrying the DNA with genetic information also, okay, for the offspring. So when the sperm and ovum fuse together, then fertilization occurs, right, at this point, and zygote is produced. So the zygote will have all the genetic information from both parents, okay, in the DNA, and this zygote will develop into the baby, right? So the baby now has the information from the parents. So DNA has been passed down from the parent to the child, and it carries the hereditary information for the production of proteins, right? So DNA is a carrier of hereditary information, which is passed down from parent to offspring in the chromosomes found in the gametes. All right. Okay. So next, what about mRNA? mRNA, what's its function? mRNA's function is to copy the genetic code from DNA, and then to help the ribosome produce the different proteins. Okay. mRNA's job is to copy the code from DNA, and then help the ribosome to produce the proteins based on the code that is copied. So let's look at the notes now. So here are the notes for the previous slide. The learning standard is justify the importance of functions or roles of nucleic acids such as DNA and RNA in a cell. So DNA is important as DNA contains genes with genetic codes or instructions found in the center part of the molecule here, carried by nitrogenous bases ATGC, that is adenine, thymine, guanine, and cytosine, for the synthesis of polypeptides and then proteins. Now, secondly, DNA is a determinant of characteristics in living organisms. And these characteristics are, for example, blood type, eye color, and height. As we can see here, there are 3 eye colors here that are different. So they are determined by the DNA in the organism. Then genes on DNA carry instructions for synthesis of specific proteins, which then determine the characteristics of organisms. So this is how DNA determines the characteristics, through the formation of specific proteins, which then determine the characteristics of organisms. Lastly, DNA is a carrier of hereditary information, which is passed down from parents to offspring in the chromosomes that are found inside the gametes, such as sperm and ovum. So a very quick revision on proteins. What are proteins? Proteins are complex organic compounds composed of one or more chains of polypeptides, as shown here. And the polypeptides are made up of about 20 types of amino acids. So amino acids are the basic units of proteins. Now, proteins form structural components of body tissues such as muscle, hair, nails, and skin. And they form new tissues, hormones, enzymes, antibodies, and hemoglobin, short form HEAH. They allow metabolic reactions to take place and regulate body functions. And they perform a wide range of functions for cells. So that's why it's very important to manufacture or synthesize the proteins, to synthesize proteins in the cells. And the code for the formation of proteins, the synthesis of proteins must be correct. And also the types of amino acids that are joined together also must be the correct types in the correct sequence. So all this depends on the genetic code that is stored in DNA. In the third part of this video, we are going to discuss the synthesis of proteins by using DNA, mRNA, and ribosomes. So we are going to find out firstly the role of DNA in the synthesis of proteins, then the role of RNA, and then finally the roles of all the 3 components here, all right, components of the cell, to find out how they work together to produce the proteins. Okay, so in this picture we see a ribosome, and this is the mRNA that is attached to it. We also see the helper molecules, the tRNA, transfer RNA, bringing the amino acids to the ribosomes so that the amino acids can be joined one by one to form a long polypeptide chain. Let's find out more. So for the synthesis of proteins in a cell, let's start off with a story. So once upon a time, there was a cell, as shown here, and the cell wanted to make a protein called protein Z, right? So he told his manager, Mr. Ribosome, to start producing the protein Z in his factory. Now, Mr. Ribosome starts to think, well, I have all the amino acids here, 20 types of amino acids, okay, and they are enough. In quantity, there are enough of them. But, what is the code and instructions? What are the codes and instructions to make protein Z? I don't know how to make protein Z. I don't have the recipe to make protein Z, right? I only have the ingredients, the amino acids. And I have my faithful assistants called the tRNA, transfer RNA, to help bring the amino acids, the correct amino acids to me when I want to use them. But where is the code? Where are the instructions to make protein Z? So the CEO of the cell, the chief executive officer of the cell, is the nucleus, all right, which is in the left part of the cell here. And he knows what Mr. Ribosome is thinking. So he says, don't worry, Mr. Ribosome. In my house or in my office, I have the keeper of all the genetic information. His name is DNA, all right? So he stored all the information for the production of different types of proteins. And he will send you the code. So now it's DNA's duty. He's been ordered by the nucleus to send the code to Mr. Ribosome. So the only way he can do it is to copy the code and then send a copy of the code out to Mr. Ribosome, because he can't come out from the office then. So he produces a molecule called the mRNA, or messenger RNA, which copies the code from DNA's body here, and then he tells RNA, send the genetic code which you have copied to Mr. Ribosome straight away. And of course, RNA, since he knows it is his job, says, yes, sir. Okay, so mRNA will come out from the nucleus, okay, through this door, and he will let Mr. Ribosome look at the genetic code on his body, which is the sequence of bases, right? And with that, Mr. Ribosome is able to produce protein Z happily, right? By assembling the right amino acids together to form a long polypeptide chain and finally to form protein Z. Now let's find out more about the genetic code on the DNA molecule. The most important part of the DNA molecule is actually the sequence of bases which forms the genetic code. For example, CAT and so forth. Now, here we see a strand of DNA where the two polynucleotide strands are no longer twisted around each other. Okay, so here we see on one side of the DNA molecule, the phosphate group and pentose sugar join together, to form two parallel chains, right? But the most important part is the center part here, which are the base pairs. One base is from each polynucleotide strand, right? Now the sequence of bases, say, found on this strand, maybe this is the genetic code, right? The other strand is only used as a template for the mRNA to copy the code. So let's say this strand contains the genetic code, right? Here. So here's an example of the part of the genetic code. Now the genetic code is written as a series of 3 bases or codons, such as CAT, and then after that maybe below there is GTA, and then further down there may be GCT and so forth. So each set of 3 bases is called a codon. And a codon consists of 3 bases, such as CAT. It actually represents a specific amino acid that must be joined to the polypeptide chain to form the long polypeptide chain. Okay, so for example, CAT is the genetic code or the codon for the amino acid histidine. That means CAT stands for histidine. Okay, so the cell will know the ribosome will know once this code is read, CAT is histidine. So histidine will be brought to the ribosome and joined to the chain, okay, to the polypeptide chain. So the conclusion is that the sequence of bases in the DNA forms a genetic code for the synthesis of proteins, right? Now this code is not just a set of 3 bases. It is a series of 3 bases, many sets of 3 bases that are arranged one after the other. And that series of bases will help the cell to choose the right amino acid to form the polypeptide, the right amino acids to form the polypeptide chain. Okay. So if you ask what is a codon, it is sometimes called a triplet code. It is a series of 3 bases in DNA or mRNA that is the code for a specific amino acid that is used to build the polypeptide chain or the protein, right? Example of a codon is AAA, adenine, 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 in RNA. So the codon is actually on DNA and RNA, but I just refer to the codon on RNA, mRNA, okay, which actually has copied the code from DNA. So AAA actually stands for the amino acid lysine, and GUA in RNA stands for the amino acid valine. So take note, RNA has this base, uracil, U for uracil, and there's no thymine there. Okay. Now AUG in RNA stands for methionine. Okay. So the meaning of a code is a system of letters with a secret meaning. So now scientists have broken the code. They know what all this sequence of 3 bases actually mean. It actually means a specific amino acid that must be joined in the polypeptide chain to build the protein polypeptide chain, or to build the protein molecule. Okay. All right. And the interesting thing is that the same codons specify the same amino acids, stand for the same amino acids in almost all species of organisms. Okay, whether humans or other types of organisms. Usually, like the codon AUG will stand for methionine, whether it's in an animal or a human being. So coming to this, let's have a look at how the codon is arranged. How is it arranged? For example, now this is an mRNA molecule. Okay, it's made up of a single strand of polynucleotides, and here sticking out here are the bases, right? So if the bases are AUG, it stands for methionine. After that, ACG, the sequence of bases, is codon number 2. It stands for threonine. Then the next one after that is UAU, is codon number 3, stands for tyrosine, and the fourth one is GUA, stands for valine. Okay, so that means these 4 amino acids will be brought one by one in a systematic way to the ribosome, and then they'll be joined together following the genetic code here on the mRNA. Now, what is complementary base pairing? So we must know this rule. The complementary base pairing rule states that in the DNA molecule, the base adenine A always pairs with the base thymine T, or the base thymine T always pairs with the base adenine A. And likewise, the base guanine G always pairs with the base cytosine C, or cytosine C always pairs with guanine G. Okay, so for example, guanine cannot pair with adenine or thymine, only pairs with cytosine. All right, that is the rule. So here we can see in this table here, the DNA base and the complementary DNA base. So DNA base is on one, maybe on one polynucleotide strand, and the complementary DNA base is on the other polynucleotide strand, as seen here. All right. So the DNA molecule consists of 2 polynucleotide strands, and the base pairs are in the center. So if the base on one strand is adenine, then the base on the other one must be thymine. And then if this thymine, it must pair with adenine. Okay, G with C and C with G, as stated here. However, in the formation of RNA, especially mRNA, adenine does not pair with thymine. It pairs with uracil, U, because there's no thymine in RNA. So here we can see, if the DNA base is A, then the complementary RNA base will be U. Now I'm going to explain this, all right, when we talk about formation of mRNA, okay, by copying the code on DNA. Okay, let's discuss this. So the first step in protein synthesis is transcription, whereby mRNA is formed by copying the code on DNA. So how does mRNA copy the code on DNA? Now this is not explained in your textbook, but I'll try to explain here as more advanced knowledge. If you don't understand, you just remember what transcription means. That's okay. So transcription means the process whereby the mRNA is formed by copying the code on DNA, which we'll discuss in the next slide too. So let's find out how the mRNA is formed by copying the code on DNA. Okay. Now, firstly, we must know that the DNA molecule consists of 2 polynucleotide strands, the yellow strand and the orange strand. They are both polynucleotide strands with their bases here that form the bonds, okay, through complementary base pairing. So there's one strand that has the genetic code, and the other strand is called the template strand. Okay, so one polynucleotide strand has the genetic code in the form of the bases. A sequence of 3 bases represents one amino acid. Okay, so this is called the triplet code, or this is called the codon, codon ATG. Another codon is CGA, which represents maybe another amino acid, right? Now, so the genetic code is ATG, CGA here, where 2 codons are shown. Of course, there are more codons involved for each genetic code, which is the code for the synthesis of one specific protein. So the mRNA is formed by using the template strand. It will be formed through complementary base pairing using the template strand, right? Now, to make the mRNA strand, first of all, this DNA molecule will unwind and the 2 polynucleotide strands will separate from each other, okay? So that the mRNA can be formed by using the template strand. So nucleotides carrying the bases that are complementary to the bases on the DNA template strand here will be brought to the DNA template strand here. So nucleotides with the complementary bases will then join together to form the mRNA strand. Okay? So if the template strand has the base thymine, then the nucleotide carrying the base adenine will be brought here. And if the nucleotide has the base adenine, then another nucleotide having the base uracil will be brought to bond with the adenine, right? And then these 2 nucleotides carrying these bases A and U will join together, and so forth. Slowly, the nucleotides are brought that have the complementary bases to each of the bases in the template strand, and these nucleotides are joined together, and they form the mRNA strand. Okay, so if you look at the mRNA strand, now after this process of copying the code, the mRNA is formed already. It will leave the DNA template strand. It will come out from the nucleus, and it will go to the ribosome, where it will be moved to the ribosome, where it's attached to the ribosome. Then the process of protein synthesis is carried out using the code on mRNA, right? Now, if you look at the mRNA strand here, it has the genetic code like for the DNA strand here, except that thymine is replaced with uracil in the mRNA molecule. All right. So now let's talk about the full process of synthesis of proteins. Right. So now we are ready to discuss the full process of synthesis of proteins, formation of polypeptides, and then proteins, right? So this process, and there's a diagram that's similar to this one on page 80 of your textbook, but the explanation is brief, and many students don't understand what's going on. So I'll try to explain it here, all right, using this diagram, which is similar to the one in the textbook. So first of all, in the synthesis of proteins, we have to know the role of DNA, mRNA, and the ribosomes, all right, in the synthesis of proteins. Now, the first step is in the nucleus, there's the DNA, right? So the DNA will first unwind. All right, let's look at this section here. You will unwind before transcription occurs. All right, so let's talk about transcription first and then translation after that. Now, in transcription, the mRNA is formed by copying the code on DNA. So mRNA is formed through complementary base pairing with this template strand, right? As explained in the previous slide. Now, you can see that its genetic code is similar to the genetic code on this DNA strand, ATG, CGA, but here the T is replaced by U. Next, the mRNA will move out from the nucleus into the cytoplasm, and it will be attached to a ribosome where translation occurs. So translation means that it's a process whereby the code on the mRNA here is used or is read to determine the sequence of amino acids that need to be joined together to form the polypeptide chain, right? So for example, if the codon or the genetic code is AUG, it represents the amino acid methionine. So methionine will be brought to the ribosome. Then the second part of the code, genetic code, is read. So maybe the second codon or triplet code is CGA, and it represents the amino acid arginine. So arginine will be brought to the ribosome by a specific molecule which we call tRNA, transfer RNA. It will be brought, and then it will be joined to the first amino acid. Then the third part of the genetic code is read. The third codon is read, right? To determine the next amino acid that's joined to arginine, and so forth. Slowly, the amino acids are assembled to form the polypeptide chain. So the chain will grow longer and longer until finally the whole polypeptide is completed. The building of the polypeptide is completed. Then this polypeptide chain may join to another one, and it may undergo the folding process to form the three-dimensional shape of this protein molecule. Okay, so in summary, in the synthesis of proteins, genetic code on DNA in the nucleus is copied onto mRNA using the process of transcription. Next, it is translated, the genetic code is translated into a sequence of amino acids, okay, at the ribosomes, and finally the protein is formed. All right, so we have to know the process of transcription and translation, at least know the definition. Transcription is a process whereby mRNA copies the code on DNA, and translation is the process whereby the code on mRNA is read to form a sequence of amino acids, and finally to form a polypeptide chain and protein. So we've already discussed synthesis of proteins in a cell, starting from the genetic code on DNA. Let's look at this picture, which shows how it occurs in a cell. Right? Now, so first you have the DNA molecule, and the DNA will unwind so that transcription can occur, whereby the code on DNA is copied onto mRNA. That is the definition of transcription. Okay, try to remember this definition. Transcription is the process whereby the code on DNA is copied onto mRNA. See, the DNA has undergone the process where it unwinds, right? It unwinds. So it opens up here, and next, the mRNA is formed by copying the code on DNA. So this is the mRNA that's formed, right? So the mRNA is formed through complementary base pairing process. Now next, that means complementary bases are used to bind to the bases on this template strand. But after that, it will come out. It won't be stuck here forever. It won't be attached forever, because it binds using the certain bonding that's very weak. So it will next come out. mRNA will come out. Okay, mRNA moves from nucleus to ribosome. All right. So it comes out and moves out to the ribosome. So ribosome is this structure here, all right, with the two subunits, the blue one and the pale yellow one. So the mRNA will sit here, will be attached to the ribosome. It will rest there, and then translation occurs on the ribosome, whereby types and sequences of the sequence of amino acids is determined using the code on mRNA. What is the type of amino acid that the cell needs, the ribosome needs to, allow to join together to form the polypeptide chain. So translation is a process whereby the types of amino acids and the sequence of amino acids is determined based on the code on mRNA here to form a polypeptide chain. All right. Now, what's the function of another RNA molecule called the transfer RNA? It is used to transfer the amino acids, the different amino acids, to the ribosomes. So you get the amino acids from the cytoplasm and bring it to the ribosome. And it has the complementary bases, compared to the codons or the triplet code on mRNA. Okay, so that it can bind to that and then bring the right amino acid, okay, to join to the others, to the other amino acids that are at the ribosomes here. Okay, now we don't have to really know the function of tRNA, so just to know that it is sort of a helper molecule that brings the amino acid molecules to the ribosome. That's all. Okay, so this tRNA. Now next, amino acids are joined to form a polypeptide chain. So here you see the circular shapes are all amino acids of different types, because different colors means different type of amino acid. So finally, as they are joined together, the chain will become longer and longer. The polypeptide chain will become longer and longer. And after that, when the polypeptide chain is completed, then it can be folded to form the protein. Okay, so that is the process of synthesis of proteins in a cell, starting from the genetic information in DNA. Here are the notes for our discussion just now. The question here, which can come out in the essay section, is explain the role of DNA and RNA in the synthesis of proteins. Okay, so firstly, we must mention that DNA contains genetic codes carried by nitrogenous bases A, G, C, T for the synthesis of polypeptides, which will then form proteins. So the genetic code is written as a series of 3 bases or codons. Okay, for example, TAC is one codon, GCT is one codon, then ATG, another codon. Then that will determine the types of amino acids. A series of 3 bases or a codon stands for one amino acid. So the whole series of 3 bases in the DNA will determine the types and sequence of amino acids in a protein to be synthesized. Now the process of protein synthesis starts in the nucleus. The DNA unwinds. Then the 3 base sequences in DNA are transcribed or copied onto mRNA. That means the codons are copied onto mRNA to form a mRNA strand with codons. And this process is called transcription, when the code on DNA is copied onto mRNA, right? So the codon here, for example, AUG, this is a 3 base sequence or a codon. It's the code for the amino acid methionine. And then after that is another set of 3 bases, CGA, stands for another amino acid, and so forth. Okay. Right, number 5. Next, after it is formed, the mRNA moves out from the nucleus into the cytoplasm, where it binds to a ribosome. Okay. This is the ribosome with 2 subunits, and the mRNA will bind to it, right? Then translation occurs on the ribosomes. So earlier we have transcription, which is the copying of the code from DNA to RNA, mRNA. Now we have translation. Well, what's the meaning of translation? Just say this word, translation, right? So it is a process in which the genetic code or mRNA codons, which consists of 3 bases, right? Codons consist of 3 bases. A codon consists of 3 bases. So translation is the process in which the genetic code or mRNA codons is translated or used or read to determine the sequences, the types and the sequence of amino acids in the polypeptide chain that is being synthesized. Okay, meaning the mRNA or messenger RNA codons are translated into an amino acid sequence. They are read and then they are used to form the amino acid sequence, which will then form the polypeptide chain, and then it will form the protein when the polypeptide chain is folded. Now, how does translation occur? So based on the messenger RNA, mRNA codons or triplet codes consisting of 3 bases each, another type of RNA called tRNA or transferred RNA will transport the specific amino acids to the ribosome to be joined to each other. So this is the transferred RNA. It looks like a conical flask. It also has the complementary bases at the bottom here. But its main job is to bring the right amino acid based on the code, the codons that are found on mRNA. Okay, so let's say the first codon here, okay, red, green, and purple one. Let's say this codon stands for the bases AUG, adenine, uracil, guanine. Now this is the code that tells the ribosome to get the methionine. So transfer RNA will bring methionine to the ribosome. Okay. Then the next code is CGA. Let's say CGA. So CGA is the code for the amino acid arginine. All right. So another tRNA molecule will get the amino acid arginine and bring it to the ribosome. So arginine will join to methionine. The two amino acids will join together. And the code UAC is the code for amino acid tyrosine, which will also be joined to arginine. Okay. So in this way, the amino acids are joined in the correct order and sequence to form a specific polypeptide chain, and then after that to form a protein molecule made up of several hundred amino acid molecules, such as these colored circles here. They represent amino acids. So here you see the ribosome, okay, in the cytoplasm. Now, ribosome can be suspended freely in the cytoplasm, or it can be also attached to the rough endoplasmic reticulum. So both these types of ribosomes, they are the same. They can carry out protein synthesis. Okay, so ribosome is the site of protein synthesis where amino acids are assembled to form a polypeptide. So it's like a factory with a assembly chain. So number 2, mRNA provides the genetic code which is translated into a sequence of amino acids. As we said, 3 bases equals to 1 type of amino acid. So the tRNA's job is to bring the amino acid to the ribosome, the right amino acid, okay, to be joined to the other amino acids that have already been produced, not produced, to join to the other amino acids that have already been joined together. So one by one, the code is read and each amino acid, specific amino acid, is brought to join to the previous amino acids. In this way, the polypeptide chain will start to form and it will grow. And it consists of a few hundred amino acid molecules. Okay. Right, so now we're going to look at the conclusion as stated in the textbook on this page. Right? We should be able to understand the conclusion here now. So the sequence of bases or nucleotides on the DNA molecule here determines the codons or triplet codes on the mRNA molecule, because mRNA copies the code from DNA. Then next, the codons or triplet codes determine the sequence and types of amino acids that are joined together to form the polypeptide chain, which is formed at the ribosomes here. Okay, so the polypeptide chains are then used to form the protein. Okay, so actually, if I were to explain this now, we can say that the sequence of bases in DNA determines the codons on mRNA, which is formed by transcription, by copying the code from DNA, right? Then next, these codons are used to determine the types and sequence of amino acids which need to be joined together to form the polypeptide chain. So this process is called translation, the process whereby the code on mRNA is used to determine the types of amino acids that need to be joined together, right? And finally, the polypeptide chains are then used to form a specific protein. Right, so that's all for this lesson. Do share, like, and subscribe. And take care. Goodbye for now.